Talk about the nature of biology today. Um, I suppose you're listening to this because maybe you missed class or you just want to go over what we covered in class. So this chapter kind of covers just some general topics about biology before we dive into the course uh, and immerse ourselves in the microscopic stuff. So you probably have a little idea about what is science and what is maybe biology before you even came to this class. Science is a process. It is not just a bunch of facts and figures. What science does is it investigates things, it tries to explain things, and it tries to make predictions about what's going to happen. So I just read this morning that the ozone hole is getting smaller now. The ozone hole was actually, we think, caused by the release of freon, which is air conditioning gas, from mostly cars, but also from spray cans back in the 60s and 70s into the air. And the freon in the air gradually made its way up into the upper atmosphere. And then, because of the way the air moves throughout the upper atmosphere of the planet Earth, the freon tended to accumulate at the poles, the north and the south poles. So at the poles, holes developed in the ozone layer, which were alarming, and they were getting bigger. In response to that, there was a huge controversy. Freons, all the air conditioning gas was changed, and a new type of freon was put in cars and home homes and refrigerators, which is much less ozone harmful. And what I've just read today is that the ozone hole is closing, and potentially by mid-century it'll be completely gone. So there you go. Science investigated the problem back in the early 70s, attempted to explain the hole in the ozone at the North and the South Pole, and then predicted that if we got rid of Freon 12 and Freon 22, which were the harmful freons, that the problem would subside, and sure enough, it did. Um, Science is not about the truth, though. It's about coming up with good explanations, which sometimes are not the right ones. The legal system works a very similar way, but do note that as of September 2011, 273 citizens of the United States were convicted uh, of crimes and sentenced to jail time, and then found to be not guilty because of the DNA evidence that was in turn examined. We will never know how many people were convicted before that because it was reasonable. It looked like they actually committed the crime and were jailed, and there was no evidence to later, free them. So biology is not just a bunch of facts and figures. It's people out there studying and trying to figure out, what makes trees grow, what makes monkeys howl, and why do fungi grow on the forest floor. And it's biologists trying to figure out what makes living things work. So the whole of science rests on what we call the scientific method, and you can read different renditions of how this method works. The one I have here is basically the way the human brain works. We tend to observe the world with our eyes, our ears, our hands. We feel things. Our nose smell things. We taste things, and our brain analyzes what we're experiencing. And then we quickly come to conclusions like, oh, I don't like this chicken. It doesn't taste good. Uh, or I do like this new app for my phone because it's pleasing to me, or whatever. Um, Very few of us actually lead into an experiment, but that's where science gets a little different. Scientists do the same thing that we all do. They observe things, they think about them, and then they come to conclusions about those things they were thinking about. But then they go ahead and do a test, and it's a simple experiment. Now, if you do enough of this process, sometimes you'll generate a theory. But the scientific method basically is a revolving or um, a repeating process where people observe, they analyze, they hypothesize, they experiment. Now that generates new observations, which then brings about more analysis, more hypothesis, and then new experiments. And in the end, what is ground out of that are theories. So you can see over here in this diagram, theories are way down here at the bottom, and they come about because of multiple experiments testing multiple hypotheses, over time. So we'll talk more about the controlled experiment in just a few minutes, but I just want to talk about some really classic experiments that come out of history. If you're going to do an experiment, uh, and you really want people to believe what you're saying, you have to um, do an experiment where you have two groups. Now, if you're a biologist, you're going to have two groups of living things. You're going to have one group of living things that get a whole set of conditions, a certain amount of light every day, a certain amount of food every day, a certain amount of water and everything. And then you're going to have another set of organisms that get exactly the same things that the first set get, except for one difference. If you're working on rats, you're going to have 500 rats and they all have the same conditions, but 250 of them have a slightly different condition than the other 250, just one thing. You can only test one thing at a time. That's the controlled experiment. The difference between the two groups is called the variable, the manipulated variable. The response to the manipulated variable, like if you were testing drugs on rats, the difference, like if 250 rats were getting just water and vitamins, and the other 250 rats were getting water plus vitamins plus one extra chemical, and then you saw some rats dying in that group, the responding variable is the mortality of the rats. So the responding variable is a little different than the manipulated variable, but the responding variable is caused by the manipulated variable, but the responding variable is what you measure. At any rate, there are two classic experiments that come out of early science. In the 1600s, Francesco Redi was disputing the whole concept that maggots come from decaying meat. Basically, he put meat into jars, and some jars he covered up with cheesecloth. Other jars he left open. He observed flies landing on the meat in the open jars. He observed flies landing on the lids of the closed jars, but he observed no maggots on the cheesecloth covered jars, but he observed maggots on the open jars where the flies were able to reach the meat. There was an old theory, it's called spontaneous generation, which came out of human experience. It was a general belief that simple life forms could arise from non-living material. And Francesco Redi is one of the first scientists that started to disprove that concept. Then fast forward to Louis Pasteur in the late 1800s, or I guess a little bit past the mid, 1861. He did a classic experiment with boiling flasks of broth. And what he did was, a lot of scientists still in the 1800s believed that diseases were caused by evil forces and that microbes could simply grow from anything. So if you got sick, microbes could just grow in you from just an evil force and take your life. At any rate, what Louis did was he put broth into these goofy flasks that had this curved top on them, and he then boiled the flasks. And what he did was he let them sit. Now, what Louis had was a set of flasks where if you have a straight top on it and dust can fall in, after you boil the broth, it spoils in a few days and it smells horrible. It rots. It's like spoiled milk that's been sitting out too long. But if you put this curved neck on it, the broth never spoils until you snap the top off the flask, and then the microbes grow inside. So this is a simple controlled experiment. He did a whole bunch of these flasks. He put together a whole bunch of the flasks, and some he left intact and others he broke off. Breaking off some flasks is a single different condition. And then he watched the broth spoil in only the flasks where the tops are broken off. Louis laid to rest the whole concept that microbes could grow from just dirt. In fact, it was only a few years later that Robert Koch Hospital is named after Robert Koch, came up with the germ theory of human disease. And so finally in the late 1800s, we began to grow microbes in culture dishes to try to figure out how to kill them and get people healthy. So I'm going to move on now to the general characteristics of living things. Living things are very difficult to define. If you look in the dictionary, there's really no standard definition of life. Some dictionaries will say, well, life is the time between birth and death. Well, that's not helpful to define things from giraffes to slime molds to mushrooms. But there are 7 or 8 characteristics, depending on how you group them, that all living things seem to have. Now, if you don't have these characteristics, you're not living. So viruses actually are not living because they don't have all these characteristics. They sure do make life miserable, but they're not actually considered living because they don't have all of these features. So number 1, they have cells in them. Okay, cells were discovered in the 1600s, and that was the beginning of developing this these characteristics of life. Number two, all living things can reproduce themselves. Number three, they're based on a genetic code. So you've got some chemical in you that stores information on how to build your body and make it work. Number four, you grow and develop. So you don't just poof into existence as a intact being. You start out small as a single cell, and then you change over time and you become another form. That's pretty typical for just about all life forms. You obtain and use materials and energy. Then we got to go get stuff. We have to eat. We use the materials to build our body, and we use the energy to make our body work. All living things, do that. We respond to our environment, meaning, is it light? Is it dark? Is somebody touching me? Is it comfortable? Is it too cold? Is it too warm? All living things can respond to the environment in some way, shape, or form. Really simple living things just chemically respond. They can't move, but they can chemically respond inside. 7, they maintain a stable internal environment. What that means is the chemistry, like inside of your blood, is a very stable thing. So we can take a blood sample from any student and see if they're healthy just by looking at the levels of all the chemicals that are in the blood, which should be within specific ranges. If they don't fall in there, then we know that there's something wrong with you. And then lastly, living things don't stay the same over time. They change. And so in the fossil record, that's what this diagram down here is showing. The horses on the Great Plains, which the Great Plains are the birth of the modern horse, started out as little Chihuahua animals that ran around, and over time, in spurts, they got bigger until they became the modern, present-day horse. So living things change over time. So those 8 characteristics, that are listed here are the 8 characteristics that kind of are used to define life. Now, most biologists define behaviors and colors and patterns 2 ways, proximately and ultimately. And you're going to hear about this throughout the course. Proximate causes for behavior and for features are always done in terms of survival, or in other words, satisfying immediate needs. Whereas ultimate causes of behavior are an evolutionary thing. They look at over the generations, not right now, but over the generations, this is how this happened. So for example, these 2 pictures here came from the Missouri Conservationist, which is a publication that the Missouri Department of Conservation here in Missouri produces, something that you should get at home if you don't. This is the caterpillar version of this butterfly. This is Missouri's largest butterfly. That's the giant swallowtail. The caterpillar looks sort of like bird poop. And the proximate explanation for the caterpillar looking like that is it's camouflaged and it helps it to survive better. It also has an appendage that can actually secrete a really obnoxious fluid. So should an animal actually start to harass it, it can actually secrete things onto the predator to defend itself. The wings, the proximate explanation for wings are it allows this butterfly to find a different food supply than the caterpillars eat mostly leaves, whereas the butterflies are sucking nectar from flowers. Butterflies fly also mainly to find mates. They live longer as caterpillars than they live as butterflies. The ultimate explanations for these 2 things are this animal evolved, it laid eggs on leaves, which are a convenient place to lay eggs, and those eggs have to grow up into something that can immediately feed on a food source. Why not eat the leaf? So this animal right here is a leaf-eating machine because this animal puts the eggs onto leaves, which then this animal eats. Over the millennia, evolution produced camouflage and defense, which keeps this animal safe from predators. Then, though, it's really hard for these types of forms to find mates. If you're a caterpillar on a leaf on a tree, you can't find another mate to reproduce. So what do? You actually, I didn't show a picture here, but you grow into a cocoon or a chrysalis, and then you grow wings and you explode out of the chrysalis, you let your wings dry, and then you fly away. The flight is actually to allow this animal to mate so it can lay more eggs and start the life cycle all over again. Most butterflies actually live longer as caterpillars than they do as butterflies. So the ultimate explanations for this animal always rest on evolution. The proximate explanations always rest on immediate needs. The animal needs to be camouflaged. It needs to defend itself. It needs to be able to fly, to feed, and to mate. But the ultimate explanations are, well, evolution produced that. Because the flying animal isn't tied down to leaves, it should then be adapted to eat other things. Whereas the eggs are laid on leaves, so the animal that immediately grows from the egg needs to be able to eat leaves. Now, those are evolutionary explanations that they came about because of the process of change over time. Now, before we get to the end here on the metric system and tools, got to talk briefly. There are a bunch of different branches of biology, from medicine to cell bio. We're going to touch on a bunch of these this year, and you should know at least these. Anatomy is the study of plant and animal structure. Physiology here is the study of plant and animal function, or organ function. Botany is the study of plants. Ecology is the study of the interactions of living things. Entomology is the study of insects. Genetics is the study of heredity. We're going to touch on a lot of these throughout the year. Mycology, won't say much about that. That's the study of fungi. There's only a few actual mycologists in the St. Louis area. They're rare, but you need them, especially for mushroom poisonings. They need to be able to. You need those kind of people to be able to identify mushrooms. Paleontology is the study of ancient life. Taxonomy is the study of classification of life forms. Biochemistry and cell bio and microbiology study little stuff. Biochem studies chemistry of living things. Cell bio and microbio study either cell qualities or lots of different microbes in living things. They're found in living things. Embryology is the study of developing organisms, like even humans. Ethology is the study of animal behavior. Hopefully you'll never have to see an oncologist, but oncology is the study of cancer. And zoology is the study of animals, and that's only some. There are others. But now I need to dive into the metric system. And we're going to talk more about this in class, so I'm not going to beat it to death here. But suffice to say that science is the reason the metric system exists. Worldwide, there's only a couple of countries that don't use the metric system nationally. There are there's two systems of measurements in the world. There's standard units and metric units. The standard units are also called British units. Most of the world has gone to metrics because it's easier to work with. And so you'll notice the United States, Liberia, and Burma are still nationally using standard units and have not switched to metric. In the United States, all medicine is done on metrics, but all consumer goods are sold in standard quantities, not metric quantities. So when we buy gas, we're buying it by the gallon, not by the liter. And when you buy meat, you're paying for it by the pound, not by the kilogram. So, but drugs, on the other hand, you're buying by the gram. If you're at the hospital, you're paying for drugs, and it's always milligrams of drug per kilogram of body mass that they're treating you with. So we have to convert between the two right now because we're not in metrics. Metrics is easier than standard because metrics is based on the power of 10. Whereas the standard units grew out of human experience. An inch was a couple of barley corn seeds. A foot is about the length of a human foot. A yard is about the length of a human, a little bit more from shoulder out, so the length of your arm. Those are great, except that science sort of demanded a standard unit of measurement that would be worldwide uniform, so that we could maybe send a person to the moon, and we wouldn't, or to Mars or something, and we'd be safe in doing it. We lost on September 23rd, 1999. The Mars Climate Orbiter was lost because a NASA ground-based computer system transmitted thrust information in pound seconds rather than Newton seconds. What happened then is the spacecraft went into a super low orbit that it could not sustain, and it crashed into the surface of Mars. That cost us $125 million, and we got no information back about the Mars, atmosphere, all because we didn't do proper conversions. So you convert all the time. You convert when you're buying food. If you're in the cafeteria and somebody's selling cookies, and it's, cookies are 25 cents, you're thinking you got $1, you can get 4 cookies. So you're going to do some conversions in here, but we don't do them a whole lot. Just for the record, you can read about this in the notes here, but the current definition of the meter is the length of path traveled by light in a vacuum during light interval of time of 299 billionths, whatever, of a second. The liter was originally a mass unit, but it's now the volume of 1 kilogram of pure water at a maximum density and standard pressure. Today, the liter is defined as basically 1,000 cubic centimeters, and the gram is 1/1000th the mass of the standard kilogram, which is somewhere, I believe, in France at the International Bureau of Weights and Measures. And this standard is about 90% platinum and 10% iridium, which are very unreactive metals. So that's what we base the metric system on. The standard system is just based on the oddest stuff, and that's why there's no consistency in converting. It's not 10, 100, 1000. It's 2, 4, 6, 8, 10, 12. It's very hard to remember all the conversions in the standard system. We'll talk more about those in class, though. Now, lab safety. Please be careful in the lab. We got Sharpies in the lab. We'll be handling razor blades and potentially scalpels during the year. When somebody's looking through a microscope, you don't want to bump them because their eyeball can hit the lens and scratch their eye. We're going to have water on the tables, and we're going to be pulling electrical cords down on the tables, and you don't want to get water spilled on cords and get somebody electrocuted. So you just got to kind of move slowly during lab and take your time, and keep things clean. The last things here are the tools that we use in biology. And really the most important tool is the microscope. Microscopes save lives. They are the backbone of hospital medicine. If you get sick, we're going to take some body fluids from you, and somebody's going to look at your body fluids with a microscope and see what you've got in you. The light microscope is different than the electron microscope. So there's two general kinds of scopes. Light scopes are much cheaper than electron scopes. There are two kinds of light scopes shown in this picture. This one is a simple one that's using sunlight to bounce off a mirror to come up through lenses. This one is our light microscope that we have in class. And these are outstanding scopes. This is an actual LED lamp housing with a switch. So the strategy here is light shines up through, this is called an iris diaphragm, which regulates the light coming up through the stage. This is the stage here. And this is where the slide will sit with the specimen on it. Then the light shoots up through an objective lens, which is either 410 or 40 power. And then it goes up and it hits a prism, which is a triangular chunk of crystal glass. That bends the light up to the eyepiece. This is another 10 power magnification, and then you see the object that was down here on the stage. This can magnify up to about 1,000 power, but in class we'll only go up into the 400s. So our scopes are outstanding. They're better than most college scopes on college campuses. The light microscope is the workhorse of nearly all hospitals worldwide. When we say, oh, we're going to send it to the lab, somebody's going to look at something with a microscope, or they're going to grow things with cell culturing, which I'll talk about in a minute. Electron scopes come in two types, TEMs and SEMs. TEMs are long tubes that shoot electrons. The tube is actually an electron gun. The electrons are focused with magnets. The tube is a vacuum. And you have a specimen down here on the bottom, and you're basically shooting an electron beam right onto a specimen. You can get much higher magnification with an electron scope. It can go up to a 750,000 or more power. And so you can see stuff like this. These are two tiny little mitochondria, which are the powerhouses of your cells. We would never be able to see this with a light microscope. So we've been able to see secrets of cells with TEMs. TEMs produce these kinds of 2D images. They're always grayscale or black and white. They're never in color because you can't see colors with electrons. Now, the SEM's a little different. They're not big tubes, they're chambers. And what you do is there is a short tube on top that shoots electrons down onto a specimen. The specimen then shoots electrons off of itself, and a receiver then interprets those electrons and produces this three-dimensional image. So you'll notice this is a 2D image. This is a 3D image. SEMs produce 3D images. The trade-off is, though, SEMs can theoretically go up to about 500,000 power, which is not as high as TEMs, but they give you this surface view, which TEMs cannot. So TEMs are excellent for looking through little thin slices, and SEMs are great for looking at things, which is how we see the world. We don't look through each other, we look at each other. So this is not how we look at things with our eyes. This is how we look at things with our eyes. So the advantages of electron scopes is they have great magnifications, and they're thousands of times better than a light microscope in terms of magnification, but they're extremely expensive. They're millions of dollars. The specimens are almost always dead, and they're very difficult to use. And on top of it, you have to have the specimen in a vacuum. So the images from TEMs and SEMs are also, remember, they're always black and white. They're never in color. Now, lastly, all labs, good labs, will have centrifuges in them. Centrifuges are just equipment that spins things at high speeds. They're like a kind of a drill motor that spins this round housing. It's called a drum. This is what happens when you spin blood. It separates into a red area, a little thin white area, and then a yellow area. The yellow area is plasmid, the thin white area is white blood cells, and the red area is red blood cells. That happens because your blood is a suspension, and the components will separate when you spin them and subject them to incredible centrifugal forces. So centrifuges are great for separating things. Microdissection allows you to see really small things and actually take them apart and put them back together. If you ever have heard about microsurgery, reattachment of fingers or severed arms, a surgeon actually uses microsurgery to do that. You can't do it with the naked eye. The nerves are too small. You actually have to peer through a microscope, and with super fine instruments, you put the nerves and the blood vessels and things back together. Very time consuming, very tedious, but it's amazing. One of the last things here, a procedure that is used in all labs around the world, especially hospital labs, is cell culturing. If you're sick, somebody's going to actually take some of that fluid they sample from you in a biopsy, and they're going to put it on a dish. Somebody else is going to look at it with a microscope, but somebody's going to put it on a dish, and they're going to handle it with gloves because they don't want to get sick like you are, and they're going to let it sit for a day, and then stuff's going to grow on that plate. And when they see the stuff growing, you can identify it. Once you know what the microbe is that's growing in you, then you can figure out what antibiotics you need to take to kill it. So just because you have pneumonia doesn't mean you're necessarily going to get better right away. We need to know what kind of pneumonia you have. And the only way to know that is to either look with a microscope, which often won't work, or grow things in cell culturing, which usually does work. So if you have any questions further about the introduction to bio, please ask in class and I can help you with them. I hope this was of some kind of help, and I'll see you in class next time.